What's up? Is this thing on? You asked a lot of questions. All right, this is the first installation of you asking questions and we gonna answer your questions. Oh, I'm looking at myself. Oh man, this is bad. Okay, so questions today for our first installment of You Ask a Lot of Questions comes from Grace. She has given us 4 vocab words: tetrads, exonuclease, multiple alleles, and telomeres. Okay, write that down. Tetrads, multiple alleles, exonuclease, and telomeres. These are somewhat related with the exception of multiple alleles. So I'm going to knock that one out first. Okay, so multiple alleles is when you have an allele is a variety of a gene, right? So we have the gene for hair color, right? But we have multiple different hair colors now. Thank goodness we got red, we got brown, we got black, we got different shades of red. And so that is what multiple alleles is, right? Okay, so the same can be said of Mendel's pea plants, right? He had the allele for height for the plant. It could either be tall or short. So there's 2 alleles, multiple alleles. Multiple alleles usually refers to when you have more than 2, okay? So like if there was tall, short, and medium, if those were the options, then you would call that multiple alleles. With our eye color, we have multiple alleles, right? Now, you can remember back a time evolutionarily where we had 1 hair color and 1 eye color, okay? That would not be multiple alleles. That would just be like the gene. The gene makes the protein that gives you color, and that's it, okay? As soon as you get 2, then you got a dominant recessive, right? And then as soon as you get 3, that's typically what we call multiple alleles, okay? This also works for human blood type because we got A, B, and O, okay? But if we just had A and O, we wouldn't necessarily call that multiple alleles. Got it? Okay. Now, let's jump into the tetrad. The tetrad is 2 X chromosomes together. Tetra means 4, okay? And so when you have your chromosomes lined up during meiosis 1, metaphase 1 of meiosis 1 is the only time that you would ever see the tetrad. You'll never see the tetrad in mitosis, and you won't see it in meiosis 2. Because in mitosis, you just have the X's. All the X's lined up in the middle, and then they go okay, all at the same time. However, in meiosis 1, we are doing a double split from quadruploid in our germ cell layers, which is the ovaries and testicles going through meiosis to create gametes, aka egg and sperm. And in order to do this, what we have is we don't just line up X, 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 and then split them apart. That's mitosis. If we're doing meiosis, what we have is we got this X lined up with another X. And then those 2 together form the tetrad. Look it up. It's tetrad, okay? The importance of the tetrad is that this is where crossing over occurs, okay? Where this X and this X lined up next to each other in metaphase 1 of meiosis 1, they might swap a section. So maybe I'll lose a little section and it'll jump over. They'll swap, okay? And most of the X stays the same over here, except a little part just got swapped. And most of this X stays the same, except for a little part got swapped, okay? And this crossing over is a source of genetic variety. Why does it happen? Random chance. Why has evolution preserved that random chance? Because it gives you more genetic variety. This is part of the reason why all of your siblings will look different. Okay? No matter how many kids your parents have, how many hundreds of kids any, insect or fish or whatever, or plant will make, because of this crossing over event that occurs in the tetrad, setup, we will have more variety, okay? It doesn't make any new genes, but it does give you new gene combos, like brown hair and red hair, or blonde hair, blue-eyed, blonde hair, brown eyes, right? So you can look at all the variety in your siblings and know that, like, well, your parents' genes didn't change when they went to make that It was just shuffled a little bit differently. So every egg your mom makes is going to be unique because of crossing over. Every sperm your dad makes is going to be unique because of crossing over. Okay? All right. Then telomeres. Telomeres are the end of the DNA. Okay? So in your chromosome setup, the very, very tip end is a telomere. The very, very bottom end is a telomere. Okay? The telomere has a protein that controls it or breaks it down called like telomerase. Okay? And telomerase works over time, and the telomere shortens over time. This is kind of like your biological clock, and it was thought for a long time to be a measure of how old you are or how healthy you are. And that research has kind of faded out a little bit because they thought, oh, this is the fountain of youth. If you figure out a way to turn off telomerase and keep your telomeres long, then your DNA chromosomes, they stay long, and then you still maintain your information and you get to stay alive forever. Turns out this is not really how it works. Typically, biology is a little more complicated than we think, and so that was a simple fix that didn't turn out to be the case. Okay? And our fourth one, exonuclease. Exonuclease is something that exits nucleic acids. Okay, so this is going to come into play when you are making DNA for DNA replication. You have to put down an RNA primer. So RNA primase comes and puts down an RNA primer, and then that allows DNA polymerase 3 to jump on and go, okay, but there's still a piece of RNA there. The rest of it's DNA. Thanks, DNA polymerase. But this part, this is RNA. Well, we don't want that because we're DNA replication. So leaving some RNA on the DNA is not where we want to go. So you got to come and you got to pull those out. You got to exit those nucleic acids. The enzyme that does that is called exonuclease. Okay? And in some things, I've actually found that that is confused with another version of DNA polymerase. Okay, now there are like 20 different versions of DNA polymerase, whether you're talking about bacteria or eukaryotes, and whether you're talking about the leading strand versus the lagging strand. And so there's actually like a bit of kind of confusion there, but so exonuclease might be called just a different version of DNA polymerase. Or there might be a DNA polymerase that not only pulls out the RNA, but inserts the DNA in its place, all in one swoopy jank, okay? And that would be a pretty fancy DNA polymerase. And right, so exonuclease might not exist just like as a single enzymatic unit. It might be combined or a subunit of a larger type of polymerase. Okay, that's it. Thanks for tuning in to your first installment of You Ask a Lot of Questions.